A very good morning to all of you. My name is Arosha Disanaka. I'm the president of the Ceylon College of Physicians. A very warm welcome to all of you in the audience. And it is my great pleasure to welcome you to the second session in the new cycle of Young Physicians Forum. Now, you all must be knowing that the IPF, the Vijayaram Prize and the Merit Awards are given at the academic sessions. So one cycle runs from October 1 year to August the second year, the next year. So one cycle was over in August and we awarded the prizes as well. And the next cycle has begun and we had the first such meeting in October. And this is our second meeting. And it is my pleasure and privilege to welcome both the audience as well as those in the judging panel. Obviously, we judge, we give marks and award a prize also. So many thanks to the judging panel as well. I have two speakers. It is a pleasure because I know them personally as well. Both of them are two very, very bright senior registrars amongst us. It is my pleasure first to invite Dr. Gayani Chamika to speak on turning the tide, how we deal with antimicrobial resistance. Over to you, Gayani, to take over and make your presentation.
Thank you very much for those kind words of introduction, dear sir. So, since cavemen to what we are today, the humans tend to draw things. What challenges them the most was among one of the topics that they preferred in their art. So what you can see in this slide is how the art is from the past to present. So 5 of the main pandemics the human race had to face. From plague, Spanish flu, smallpox, cholera, and the latest, COVID-19. So it has been a constant battle between microorganisms and human, and most of the time in the past, the microorganisms won, until the day that these 3 gentlemen won a Nobel Prize for invention and purification of penicillin. Since then, antibiotics were quite successful for several decades, but the antibiotics were mainly having a scorched earth approach, which is the antibiotics mainly aim to either kill or inhibit the growth of organisms, and by that it introduced a strong selective pressure for the microorganisms to become resistant. Apart from this, because of the broad spectrum activity, antibiotics obliterated the protective normal microbial flora as well. But people tend to have faith on antibiotics and other antimicrobial agents from the very beginning. And in the Second World War, this was one of the taglines: thanks to penicillin, he will come home. But the inventor himself, Sir Alexander Fleming, had his doubts from the very beginning. Even in his Nobel Prize acceptance speech in 1945, he mentioned that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. And so as the physicians today, one of the main challenges that we have to face is tackling the antimicrobial resistance. So how are we going to turn this tide is the question. So the outline of my speech would be: what is antimicrobial resistance, the sources of multi-drug resistant organisms, the mechanisms acquired by organisms to develop multi-drug resistance, trends and burden of human infections for resistant organisms, and strategies to contain the resistant, and the treatment modalities. So the ability to survive the exposure to an antimicrobial agent by a disease-causing microbe is called as resistance. And this ability is developed against a particular antibiotics which was previously considered to be an effective treatment. And the global collection of these resistant genes in both clinical and environmental samples are called as the antimicrobial resistome. So as we know, antibiotics mainly target specific functions or structures of the bacterial cell, mainly cell wall, folate synthesis, cell membrane, protein synthesis, DNA and RNA synthesis. By applying a specific pressure, the antibiotics target to damage or kill the organism. To withstand this, the microorganisms have also developed mechanisms, mainly if an antibiotic is targeting a cell wall, a mechanism has developed to efflux the particular antibiotic. And there have been modifications in the target, and also they have developed mechanisms to bypass these pathways, and also they have developed enzymes which can inactivate the particular antibiotic. For example, we see a lot of reports saying extended spectrum beta-lactam is producing microorganisms or E. coli. So this slide would show the timeline of antibiotic development and evolution of antibiotic resistance. As you can see, as soon as antibiotics were invented, the resistance has followed it. For penicillin, it was marketed in somewhere around 1943, and the resistance has developed somewhere in 1946. That is true even for the newest antibiotics we use, such as linezolid and daptomycin. So as we know, the antibiotics are predominantly derived from nature, and they have been around here for millennia. So the microbes themselves have developed mechanisms to thrive against these competitors, and they have developed mechanisms to develop resistance. So the danger is the antibiotic resistance is already widespread in nature, and there is development of resistance to antibiotics which are not even invented yet, because mainly both of the antibiotics and the microorganisms share the same environment. And the antimicrobial resistance can be transmitted either vertically from generation to generation and horizontally from in the same generation by bacterial transformation, transduction, and conjugation. And it has become a global threat that over 2.8 million antibiotic resistant infections occur every year in the USA, with over 35,000 resultant deaths. As the prevalence of resistance continues to rise, the danger is so many life-saving surgeries, organ transplantations, and chemotherapy that rely on prophylactic antibiotics will become progressively more dangerous. And if I take you in a time machine to year 2050, you would see that deaths due to antimicrobial resistance would rise up to 10 million, beating deaths due to cancer, cholera, diabetes, road traffic accidents, and tetanus itself. And these deaths would be mainly seen in the so-called developed world, and there would be less number of deaths due to antimicrobial resistance in Africa and sub-Saharan Africa and Asian countries. To tackle the antimicrobial resistance, it's important to understand some concepts. WHO has introduced this One Health concept where it says that all the things in the environment is connected, like the human health, animal health, and environmental health. Everything is connected. So the pharmaceutical industry produces antibiotics, and as doctors, we believe that these antibiotics goes to people in the clinical setting, and we can't prevent that some of it would be excreted to environment. But majority of the antibiotics that are used, that are introduced by the pharmaceutical industry is being used in agricultural industry as well. And so we can't talk about antimicrobial resistance without understanding that the antimicrobial resistance is connected to all human health, animal health, and environmental health. So to tackle the antimicrobial resistance, there have been a 10-front module introduced by the WHO in year 2019. And it says that we should lower the demand for antimicrobials and reduce the unnecessary use. For this, public awareness, the proper sanitization practices, and limiting antimicrobials in the agricultural industry, as well as the use of vaccines and rapid diagnostics, are extremely important. It's surprising to see that almost 60% of potential decrease in the number of cases of water and sanitization related diarrhea being treated with antibiotics can be reduced if we just supply people with properly sanitized water. And it's important to identify the depth of our problem by having a proper surveillance system locally, nationally, and globally. It's extremely important to go for new and rapid diagnostic tests, which would optimize treatment, because most of the time we tend to empirically diagnose patients and start treatment. And the traditional diagnostic test usually takes around 2 to 3 days. And if we have a rapid diagnostic test to detect antimicrobial resistance, it would optimize the treatment, and the treatment can be offered to the patient quickly. It would be surprising to know that even in USA, out of 40 million people who are given antibiotics for respiratory illness annually, out of all these people, only 13 million people would receive it accordingly. Other 27 million of people who receive antibiotics get it unnecessarily. This would obviously tell how important it is to have a rapid diagnostic test. And also, most published papers provide evidence to support limiting use of antibiotics in agricultural environments. It's important to invent new vaccines and to increase the coverage of vaccines which are already available. For example, by making the pneumococcal conjugate vaccine available universally, it would reduce 47% of the use of antibiotics. We have some good news as well. The vaccine for malaria is being used in Africa now, which is an important step both for the management of malaria and to reduce the antimicrobial resistance. It's important to have alternative products which can tackle infections apart from antibiotics, such as phage therapy, lysins, antibodies, probiotics, immune stimulation, and peptides. As we reduce the unnecessary use, it's almost important to increase the supply of antimicrobials to keep up with the rising resistance. And we need to use the existing antimicrobials better. For example, in a study in 2012, a randomized control study, showed that 2/3 of selected antibiotics were not available in more than half of the countries included in the study. And another study, a multi-center study done in 2015, shows that almost half of the children and newborn in the sample were treated with suboptimal doses of commonly used antifungals, which obviously can be prevented, and we need to use the existing antimicrobials better. And by having a market entry reward, they can improve the registration and distribution of new antibiotics. It's important to remember our basics, and more than half of the antibiotics prescribing for selected events in hospitals was not consistent with recommended prescribing practices according to new CDC data. So it's extremely important that we optimize our antibiotic selection, reassess the antibiotic treatment when the results of diagnostic testing are available, and use the antibiotics for the shortest effective duration. As we use the available antibiotics properly, why not new antibiotics? The story is that the pharmaceutical industry are rapidly moving away from developing new antibiotics, and they are giving up their antibiotic programs because mainly they are targeting the chronically administered medications because they are far more profitable than antimicrobials. But despite that, there have been some new FDA-approved clinical trials which are ongoing, and some have completed phase 2 and 3 trials and FDA-approved for use, such as daptomycin, eravacycline, and deflufloxacin. And there have been updates on eight of the new antibiotics against multi-drug resistant gram-negative bacteria as well. And there have been an interesting phenomena called Trojan horse antibiotics. If you remember the story of the Trojan horse, you can understand this easily. The new antibiotic, cefiderocol, is actually an injectable cephalosporin that combines catechol type siderophore and cephalosporin core with the side chains similar to cefepime and ceftazidime. And since this contains iron, the gram-negative organisms, which usually loves iron, take up the antibiotic better, which can enhance treatment of gram-negative infections caused by gram-negative organisms. When we talk about the antimicrobial resistance, it's extremely important to talk about tuberculosis because it's one of the chronic diseases which needs a long-term treatment, which can contribute to antimicrobial resistance as a result. And better diagnosis, awareness, and treatment for TB would obviously save numerous lives. And over the next 10 years, it's estimated that 770,000 lives can be saved if these better diagnostics and treatments that is used in the management of tuberculosis. And FDA has recently approved pretomanate, which is which can be used for management of extensively drug-resistant tuberculosis in 2019. But if we consider the cost, tackling the antimicrobial resistance would cost a lot, and it would be estimated as up to $40 billion per decade. So what are the new developments in alternative therapy? We have antibiotics, but are there anything new? So if you see this list, you can see that there is an exhausting list, which usually means if we have, for example, for diabetes, we have insulin and we know that it's effective. But when there are several treatments coming up for one disease or one condition, we know that usually it means that none of them have enough evidence to be used as a treatment. So if we consider a few of these things, we'll see the probiotics, which we know better. And probiotics are defined as live microorganisms which, when administered in adequate amounts, confer health benefit to the host organism. And it's considered likely that defined mixtures of bacteria or the use of non-toxin spores of C. difficile will provide therapeutic and prophylactic therapies that will improve on current clinical practice for the treatment of antibiotic-associated diarrhea. But Lancet has published its data in 2013 and concluded that there have been no evidence that a multi-strain preparation of lactobacilli and bifidobacterial was effective in prevention of antibiotic-associated diarrhea. But saying that there is still a place for fecal transplantation and other uses of gut microflora. If we consider the immunotherapy in the face of the looming antibiotic-resistant crisis, there are preclinical data rapidly emerging on immune checkpoint inhibition, modulation of cytokines and cellular therapies, but clinical trials are urgently needed to determine the effectiveness and safety of utilizing immunotherapy. Checkpoint inhibition is an interesting thing where checkpoint inhibitors have revolutionized cancer treatment and was recognized by the 2018 Nobel Prize for Medicine for the discoveries of CTLA4 and PD-1. And checkpoint inhibitors are monoclonal antibodies that block the interaction of these checkpoint molecules with the ligand and restoring immune function. So the scientists thought that because the microenvironment of both the tumor microenvironment is similar to the bacterial induced granulomas, probably this can be used in infection as well. So the MAP therapy is or checkpoint inhibition has shown that it would enhance immunity and it would enhance the lymphocyte function as well, and by that it can be used as an alternative therapy to antibiotics. There have been several monoclonal antibody therapies in various stages of researchers' development and approval, and beslotozumab is FDA approved in 2017, and obinutuzumab, which is used in the treatment of inhalational anthrax, is FDA approved since 2017. And also nivolumab is fully human monoclonal immunoglobulin 4 antibody to PD-1, and it was used in a population with sepsis, and there have been no safety concerns, but efficacy and safety studies were warranted according to a randomized controlled trial. And beslotozumab for prevention of recurrent C. diff infections have it was shown that among participants receiving antibiotic treatment for primary or recurrent C. diff infections, this monoclonal antibody was associated with substantially lower rates of recurrent infections and having similar safety profile when compared to placebo. And cytokine therapy is another interesting thing. Modulating cytokines are important immune signaling molecules that regulate or activate signaling involving the development of inflammation, cellular and humoral immunity, immunosuppression, and hematopoiesis. And this type of immunotherapy intervention has been used successfully in the clinic for treating and managing the range of diseases and can be used either in the form of infusion of recombinant cytokines or inhibition of specific endogenous cytokines. And we have seen tocilizumab, which was widely used in the COVID-19 pandemic. And the data says that it prevented the mechanical ventilation, the composite outcomes of mechanical ventilation or death, but it did not improve the overall survival of the people who were receiving tocilizumab. And there are a list of cytokine inhibitors on the play. But if we go for oligonucleotides, these are short synthetic nucleic acid sequences that may be used therapeutically to silence the expression of deleterious genes. And so by that, it can change this RNA to no disease-associated protein-producing signals, and it is postulated that this can also be used as an alternative to tackle infections. And adaptive cell transfer is that immune cells can be engineered and infused into patient to fight diseases, and CAR T cells are the most common form of cellular immunotherapy. And there have been phage-based therapy as well. And lysins and phage lysins can be are enzymes that used by bacteriophages to destroy the cell wall of a target bacterium. And there have been emphasis on lysins active against gram-negative pathogens, which would be beneficial. And the bacteriophages that infect and kill bacteria have the potential to replace antibiotics for some indications. And there are two types: the wild type bacteriophages and genetically engineered bacteriophages, which were tested by randomized controlled trials for their uses. So if we compare these things, these alternative therapies have their advantages and disadvantages. The major disadvantages would be the cost and the difficulty in their production, but they usually suppress common cell bacteria and reduce the drug discovery time, and it has wide ranges of application, and it needs infrequent dosing. And these are the main advantages of going for some things like phage therapy, antimicrobial oligonucleotides, and monoclonal antibodies. So in summary, antimicrobial resistance is a global threat, and One Health is an integrated, unifying approach that aims to substantially balance and optimize the health of people, animal, and health ecosystem. And WHO has created an action plan with 10 fronts to tackle antimicrobial resistance. And there have been new antibiotic development in the recent years with risk of developing resistance. And vaccines are still important, and preclinical data are rapidly developing in the field of immunotherapy to tackle antimicrobial resistance. So with that, I thank you for your patience listening, and I wish Dr. Dilum good luck in his presentation.
Thank you, Dr. Chamika, for the eye-opening lecture on antimicrobial resistance.
can I start questioning? Yeah, Dr. Chamika, thank you very much for that very comprehensive and eye-opening lecture. Now, what is the Sri Lankan scenario on antimicrobial resistance? Do you have any studies? Where are we?
Thank you very much for that question, sir. Actually, Sri Lanka College of Microbiologists have published some data also regarding antimicrobial resistance, and there have been several articles and publications on detection of antimicrobial resistance in the Sri Lankan setting. But unfortunately, regarding these alternative therapies and targets, we don't have any data because of the financial concerns.
One more question. You said the deaths due to antimicrobial resistance will be higher in the developed world compared to Africa in 2050. What is the reason for this?
Thank you very much, sir. The main reason would be Africa and some of the Asian countries. The main problem is the scarcity of the antibiotic, availability of antibiotics. When it is less available, I mean, probably the development of resistance is less. When in countries which uses antibiotics, probably overuse antibiotics even for the production of this agricultural industries would be affected more because of this.
Okay, thank you very much.
Thank you, sir.
I'm Dr. Nandana. Can I ask a question, Dr. Chamika?
Sure, sir.
Thanks for your presentation. Very clear, simple, and very understandable presentation. Nice. And now when you talk, do you consider this anti-protozoal and antiviral resistance also? Now they are talking about this artemisinin resistance for the malaria as well. Do you think about that part also?
Yes, sir. I'm sorry, I didn't hear the first part of your question, but I think
when you talk about the antimicrobial resistance, the protozoa and the viruses are not taken into account, or you take into that account and you think about that. As example, now malaria, the artemisinin resistance is coming up. So what do you think about that?
thank you very much, sir. It was actually considered, but due to the time factor, I couldn't include many slides which may describe it. But that's why, I mean, there is a now they are shifting more towards anti-malarial vaccines and things as well. Of course, there are resistant cases and many articles and data coming for anti-protozoal agents and antifungal agents, antiviral agents, all these things.
Okay. Okay. Thank you.
Thank you, sir.
Thank you, Dr. Chamika, and thank you for the judges for the question and answering session. So now, Dr. Chamika, you will be receiving an e-certificate for your excellent presentation, and it will be displayed on the screen. So we will move into the next topic.